PRC in Jakarta, Indonesia. So today we all be going to hear from Dr. Lu Bingxin. The title of our talk is Dynamic Modeling of Chromosomal Instability in Somatic Genome. So I would like to read out her impressive profile. So Dr. Lu is currently a Surrey Future Fellow at the Section of System Biology and Surrey Institute for People-Centered AI, University of Surrey. So she's previously a postdoc at the Department of Cell and Developmental Biology, University of College London. And she used to be a postdoc at Genome Institute of Singapore, and she got her doctoral degree in computational biology at the School of Computing, National University of Singapore. Dr. Lu, are you around?
Yes. Thank you
Okay.
for the introduction.
Yes, please, time's yours.
Yeah.
We're going to have about 45 minutes here, Dr. Lu, and please excuse me if I have to remind you of 5 minutes before time is up, so we have enough time for a Q&A session. Thank you.
Yeah, sure. Okay, so I can share my slides. Can you see my slides?
Yes, perfect.
Okay. Yeah, okay, so I will start now. I'm glad to present my work on dynamic modeling of chromosome instability in somatic genomes. Thank you for the invitation and this opportunity. As you may know, in a normal human genome, there are 23 pairs of chromosomes indicated by the different colors here. However, in a cancer, this is called the karyotype. However, in a cancer genome, chromosomes are often unstable, showing high rates of segregation errors during cell division, known as chromosome instability, or SIN for short. This can generate numerical and structural chromosome alterations, such as translocations between different chromosomes, chromosome gains, or chromosome losses, and also some segmental level gains and losses. SIN play an important role in cancer evolution. The numerical SIN can generate copy number alterations and aneuploidy. The structural SIN can generate structural variants and CNAs as well. These alterations generate heterogeneity, upon which natural selection acts to enable genomic plasticity. It can also cause cellular stresses and inflammatory signaling. As a result, SIN facilitates several processes, including metastasis, where cancer cells spread from primary location to distant tissues, immune invasion, where cancer cells acquire the ability to invade immune recognition, and therapeutic resistance, in which a population of cancer cells may expand after treatment. With more and more sequencing data, SIN has been found widely prevalent in many cancers. Here is an example. Can you see my pointer? Okay, can you see the pointer?
Yeah, I can see it moving around. Yeah, very good.
Yeah. Okay, so here is an example showing the number of gains and losses across different chromosomes in gastrointestinal cancer. You can see there are lots of copy number changes. Given the wide prevalence and importance of SIN, there have been lots of studies on SIN, starting as early as 1902. There are basic research studies, and also there are some clinical trials. And with genome sequencing data, there are also lots of data analysis. projects. However, we still don't fully understand how sin generates and what's the rate of sin and how it interacts with natural selection. This cannot be resolved by experimental approaches or methods inferring evolutionary histories backward in time. So to address this issue, we try to use a forward stochastic modeling to model the data and then use statistical inference to fit the model to data. In this study, I would like to introduce our approach in modeling copy number alterations and structural variants, respectively. First, I will show you our work, our general modeling approach. As cancer cells can either divide or die, we use a stochastic branching process to simulate tumor growth. We start from a normal cell, and during cell division, we can introduce some mutations, such as copy number alterations or structural variants. And the cells with certain mutations may have fitness advantages or disadvantages, measured by a selection coefficient. If it has fitness disadvantages, it may die. If it had advantages, it may expand. We can simulate this process until we reach a certain number of cells. Then, using the simulated data, we can apply a statistical approach called approximate Bayesian to fit the model to real data. Simply speaking, we can sample parameters of the model from prior distributions, and then we simulate the model according to the branching process model to generate some data. Because usually it's a bit hard to compare the full data, so we derive some summary statistics from the data, and then we compare the summary statistics of the simulated and the real data. If their distance is smaller than a threshold, we accept the proposed parameters. We iterate this process for many times. As a result, we can generate the positive distributions of the parameters. We can do this process and different models of evolution, such as a neutral model or a model with selection. Then we can use some criteria to select the model best describing the data. Using this approach, we can measure the uncertainty in the data with statistical distributions. Next, I will show you how we apply this approach to model copy number alterations and structural variants. First, let's say I work on modeling copy number alterations. Using live cell imaging techniques, researchers have observed chromosome segregation errors. For example, here, wait a minute. Yeah, here in this short video, you can see during the cell division, there is a lagging chromosomes. Yeah, shown here. And in another video, you can see there is a very weak chromatin bridge when the cells divide. Based on these imaging techniques, our collaborators further developed 3D lab-seq techniques, including both the imaging of cell divisions and the whole genome sequencing of single cells in the patient-derived tumor organoid. Here you can see the detailed cell division size up until around 5 days. You can observe lagging chromosomes or chromatin bridges. And more interestingly, you can map these segregation errors to the copy number profiles of the single cells. Here you can see reciprocal chromosome gains or losses, or arm level gains or losses. They correspond to lagging chromosomes or chromatin bridges, respectively. Using the data, we developed a branching process model. In total, we have 4 image trees along with their single cell sequencing data. So we use this data trying to learn some important parameters of the selection rate and the selection forces. This is our model. So starting from the tumor cells, which are already aneuploid, we can further simulate chromosome level or arm level copy number alterations. Because we didn't observe many cell death events, we only assume cell division here. We can simply model under neutral evolution, namely all cells have the same fitness advantages, and we have another model of selection in which cells with certain copy number alterations may have fitness advantages measured by this formula. We can derive summary statistics from both the copy numbers and the cell lineage trees to summarize the data. Using this model and the approximate Bayesian approach I described previously, we can infer the positive distributions of the copy number alteration rates and cell division rates. In addition to the 4 data sets with cell image trees, we have 3 other data sets with just a single cell sequencing data. So we only infer their CA rates. If we add up the chromosome level and arm level rates, we can get the total CA rate, which is about 0.2 per cell division, which means 1 CA per 5 cell divisions. This is relatively high, and this is also consistent with the estimations from another study of using breast cancer cell line data. They suggest the average rate is about 0.24 CNAs per cell division. So this suggests CNAs. This suggests a quite high CNA rate in general. I want to clarify that those estimations are based on the assumption of neutral evolution. However, we suspect there may be negative selection because after the segregation, missegregation, the cell cell types show lots of alterations that are expected to be disadvantages to the cell survival. If there's negative selection, the birth rate after missegregation is assumed to decrease. For example, suppose the normal birth rate is 0.65 after missegregation. If there are negative selection, the missegregation may decrease to a smaller value. To validate this, we use model selection. We use a criteria called DIC, which can measure both the goodness of model fit and the complexity of the model. We run our our modeling approach and both selection model and neutral model. Here you can see the DSA values for neutral model is smaller. The smaller value indicates the model fits the data better. So using these four individual data sets, we didn't find the evidence for negative selection. However, we think this may be because of the limited data size. So we try to aggregate all the data size and use likelihood ratio tests to look for evidence of negative selection. We have the null hypothesis of constant birth rate and the alternative hypothesis in which the birth rate will change to a different value after missegregation. And using this test and the aggregated size, we find a significantly a difference before and after missegregation. For example, here, you can see the estimated cell cycle time increases significantly after missegregation. And in a brief summary, here we use our stochastic modeling approach to show that the new karyotypes after cell missegregation show a relatively high CA rate for cell division, and they may experience neutral or subtle negative selection. This concludes the work on copy number alterations. Next, I would like to show you our work on structural variants. Different from copy number alterations, which only include copy number gains or losses, structural variants can be very complex. They can range from simple deletions to very complex events like chromosome, in which the chromosome is shattered into pieces in a catastrophic mutational event, and these pieces may be lost and rearranged. Some of the pieces may form extra chromosome circular DNA, or ecDNA for short. And the stretch variants may also affect copy numbers. If there is a deletion, there will be copy number loss, and if there is duplication, there will be copy number gain. So actually, copy number alterations and the SVs are closely related. Although there are so many different types of SVs, they are often caused by a few mechanisms, which is often DNA damage and erroneous repair. There are two major mechanisms. One is non-homologous unjoining. This requires no sequence homology, as the name suggests, but it's also easy to have errors during the repair, and these errors will cause structural variants. In another mechanism called the homologous recombination, it requires sequence homology during the DNA repair. It is more accurate, but sometimes it can also cause rearrangement or structural variants. There have been lots of studies on the different patterns or types of SVs, but we still don't know how the mechanisms of the SVs directly contribute to the patterns we observed. So in our work, we want to use our modeling approach to try to understand the underlying processes that can relate the different SV types, because they may be generated by the same processes. We developed a quite conceptually simple, but unifying framework to model the SV generation process. To do this, we consider the cell cycle. We introduce DNA double-strand breaks during the gap 1 phase, and we replicate the genome during the synthesis phase and gap 2 phase, and also we simulate cell division during the mitosis phase. After DNA double-strand breaks, we can repair or rejoin them based on their genomic distances. We allow some unrepaired double-strand breaks. This imitate checkpoint errors. We incorporate this cell cycle size into the branching process as used for modeling the copy number alterations. And when we simulate cell division, there's a number of centromeres on the chromosomes. If there's one centromere on chromosome, they are distributed evenly to the dot cells. If there's no centromere, they will be randomly distributed to dot cells. And if there are more than one centromeres, they will form, they will break into pieces, and this will cause a chromosome fusion during genome replication, which I will describe in more detail soon. This is an example showing the accumulation of SVs from a normal cell using our model. You can see the introduction of structural variants and the change of copy numbers on chromosome 1 with 5 double-strand breaks per cell cycle. Using our model, we can explain the formation of complex SVs. For example, here, we introduce one double-strand breaks on this chromosome, and during genome replication, these two chromosomes are fused because they lack telomeres, and in this case, they will have two centromeres. Because they have two centromeres, they will be broken during cell division. And when they broke, there can be either a very simple break, just like this. And in this case, it may cause some staircase focal duplications or serrate structural variation. But when the breaks, there may also be local fragmentation. In this case, the chromosomes will be shattered into many pieces, causing chromosynthesis and seismic amplification of very lot of amplifications. I will focus on this case in the next few slides. There's a recent study showing that complex SVs can be generated from one cell division area. For example, here you can see in this cartoon you can see the bridge formation during the first mitosis, similar to here. And this breakage will cause a chromosynthesis or defective replication, like what I show here. And then in the next mitosis, there will be more chromosynthesis and defective replications and also micronucleations causing lots of SVs. We want to use our model to recapture this phenomenon. So we simulate our model, and we use a model to simulate the cell growth until 100 cells and neutral evolution. We introduced one unrepaired double-strand breaks. And if we leave this one double-strand breaks and let it go through the cell cycle and go through the bridge formation and breakage process and also local fragmentation, we can simulate lots of copy number alterations across different cells. Yeah, this is a copy number heat map for the cells. For this cell indicated by the red arrow, you can see some oscillating copy number changes, which is showing more detail here. You can see the oscillating change of copy number 3 and 1. This is a typical pattern of chromosynthesis. So using our model, we can generate chromosome-like patterns. And we can also simulate extra chromosome ecDNAs, namely those DNA without centromere, and they will form a circle during the rejoining of double-strand breaks. Here you can see there are the slightly more ecDNAs and a positive selection, but the size and the maximum and mean copy number seems similar. This is probably because of the strength of selection is not very large. And we also want to explore how our model can generate complex SVs in either 1 or 2 cell cycles. So here we simulated 3 cases, like one cell cycle with double-strand breaks in the first cycle, and 2 cell cycles with double-strand breaks in both cell cycles. Here I mean new double-strand breaks. There will still double-strand breaks during the breakage. And then we counted the number of chromosome synthesis ecDNAs and seismic amplifications. We tested different parameters, like different double-strand break DSB rate, like 10 DSBs per cell division or 30 DSBs per cell division. We also explore different numbers of double-strand breaks during local fragmentation. Here you can see there are more chromosome fusions or chromosynthesis and ecDNAs in 2 cell cycles when there are more DSBs and more DSBs in local fragmentation. This suggests that we can indeed simulate or generate complex SVs in just 2 cell cycles. And even with 2 cell cycle with only DSBs in 1 cell cycle. Yeah, these are just simulated data. Next, we want to show the utility of our model in fitting real data. In a recent study, the researcher generated a lot of single cell data for cancer derived xenografts and also cell lines with TP53 and BRCA1 or 2 gene knockdown. We use this data and trying to fit our model on this data. For the cell line data, there are 7 data sets with different combinations of gene knockdowns. And some of them are polyploid, namely they have their genome duplicated or have whole genome duplication or whole genome doubling. For the patient-derived xenograft data, they are grouped into different signature groups. One is enriched in fold-back inversions and cis-NU1 amplifications, and for this group, the SVs may mostly caused by non-homologous unjoining. And there are also two other groups which are enriched in small tandem duplications or large tandem duplications. And these SVs may be mostly caused by homologous recombination indeficiency. Because in our model we didn't consider sequence homology when we repair the double-strand breaks, so we most focus on FB1 data size. Then using the approximate Bayesian approach, we inferred the posterior distributions of important parameters, including the DSB rate for cell cycle or cell division, and also the fraction of unrepaired double-strand breaks and the probability of whole genome doubling per cell. Here you can see high heterogeneity across data sites. And interestingly, we can also infer the posterior predictive distributions of some unobserved variables, such as the mean number of chromosome fusions per cycle and the mean number of ECDAs per cell. It is feasible to detect ECDAs from the sequencing data, but currently it's mostly from bulk sequencing data. So yeah, so I think the prediction of these unobserved variables may provide some interesting information on the data sets. And these results are also consistent with known observations. For example, for this SA118, in the original paper, they found there's a breakage fusion bridge cycle, indicated by the cartoon here on chromosome 3. So this suggests there's one chromosome fusion in this data set, and here in our prediction, the value is also around 1 or 0. So it confirms our prediction is reliable. We also found the pairwise correlations among the important parameters. Here you can see the DSB rate per cycle, the mean number of chromosome fusion per cycle, and the mean number of ECDs per cell, and the all significantly increase with the probability of whole genome doubling per cell. And there's also correlation between the mean number of chromosome fusions and the mean number of ECDAs per cell. This is because after chromosome fusion, there's usually bridge breakage, and during bridge breakage, there will be some small chromosome fragments without centromere formed, and then they will form the circular ECDA. There have been no previous report on the direct correlation between whole genome doubling and the number of chromosome fusions and ECDAs. There are only some studies implying that the whole genome doubling is correlated with CCNE1 amplification, and CCNE1 amplification is frequently targeted by BFB cycle, and this BFB cycle may lead to chromosynthesis and ECDAs. So our modeling helped to find some new correlations between these events. In addition, given that the CCNE1 amplification is also important in the formation of large tandem duplication, it seems a very important target in clinic. As a brief summary, here I have shown our model of SV generation, and by fitting to real data, we can recover K parameters in SV generation, and also can reveal some novel correlations among the SVs. and our model may serve as a foundation model to study the formation of ice. And as a brief conclusion, I have shown our approaches on modeling chromosome instability in an evolutionary framework. Given there's more and more cancer genomics data and insufficient understanding of the evolution of chromosome instability, our approach provide a good strategy to quantify chromosome instability in an evolutionary context. And finally, I want to thank my supervisor and my previous collaborators for this work, which has been mostly done at University College London when I was a postdoc, and currently I have started my new position at Surrey, and I will be recruiting PhDs and postdocs. If you know anyone interested, please let me know. And yeah, so that's all. Thank you for your attention.
Great. Thank you very much, Dr. Lu. I think it's very informative for a sloppy molecular biologist like myself. I think it's being inundated with lots of mathematical model. Interesting. Okay, so I guess the advantage of being a moderator, I got to ask question first, yeah. So and then I'll probably let all our audience. We have about 34 audience here that maybe we can maybe raise hands or you can put your question in the chat. But let me ask first, Dr. Lu. You know, when it comes to this dynamic modeling, right? And you mentioned the evolutionary processes, but I guess I'm missing the part of the selection pressure. What is the what keeps the cells to keep on going, changing its genome? I mean, you've shown that the numbers of the structure is probably different, the copy numbers is different, but I would think it should have some incentive to do it, right? Otherwise, why the cells actually carrying a risk of actually keep on getting more unstable, especially when you model it probably in vitro, right? Not so much in vivo. So how would you explain this kind of, I don't know, counterintuitively? Because you know, when it comes to evolution, you always talk about variations, but also the selective pressure. But here in this case, I don't see the selective pressure, why the cells want to keep on going and keep on changing its genome.
Okay, yeah, a long question. Okay, maybe I can go back to my slides. Maybe I can use this slide. Can you still see my slide?
Yes, yes.
Yeah. So you are asking about this selective pressure. And I think can you briefly summarize your question? It's like
Oh, okay.
you,
Right. So I came from my background, actually, I'm helping clinicians how to understand resistance, right? So we know, like you mentioned about the BRCA cell line, you mentioned BRCA1, BRCA2, and also in contraction P53. We know that those cells with the BRCA mutations, they eventually has lots of structural variants. There's lots of heterozygosity. And collectively, it's called HRD. I think it's mentioned in your slides, right? This is called homologous recombination deficiency. And this phenotype, this huge huge instability actually is a target for a drug, right? The PARP inhibitors. So the question is, I would understand that if you add the drugs, I'm kind of wondering if you have the model. So you grow
mm,
these cell lines, right? It has a
mm,
BRCA1 and P53. And then
mm,
you compare two kind of scenario, one without the
mm,
drugs, and the other
mm,
one you give the drugs, the PARP
mm,
inhibitors. And then maybe you
mm,
can create a model because what
mm,
happens is it's going to be very useful for the physicians to actually predict what to look for, which patient will eventually go to relapse, right?
mm,
Because currently we only look at maybe one reverse mutation in BRCA that actually
mm,
rescue the HRD phenotype, so we just no longer the drug's no longer effective against these particular cancer cells. So when I go through to your presentations,
mm,
I don't see the pressure. What is the selection pressure? I mean, it looks like you're just growing the cells. I mean, happily in incubators, but then they keep on actually evolving. But evolving, what are the benefits of evolving without a certain incentive to go against pressure, right? So the pressure I'm talking about, for example, is the adding the drugs, but without giving any pressure, why the cells are keep on evolving? What is their benefit of doing it? You understand what
oh,
my
oh, I
my
see, I see.
yeah,
Yeah, yeah. Okay, you are asking about this selection pressure in the data and probably in the model.
right.
Mm. So, yeah, I think it's still yeah, I think it's a bit hard to quantify the selection pressures. From my understanding, the data is probably that this um the new types with new uh copy number alterations or structural variants, they may affect some important cancer genes, such as some oncogenes or tumor suppressor genes. And for example, they may amplify some oncogenes, and this will increase their fitness advantages. So in that case, the cells may grow, and in some case, yeah, actually in some cases, I think these new types may be disadvantages to the cell survival, but then they will die, but we cannot observe them.
Mhm.
Mm. In in our model on the structural variants, actually we currently we incorporate a very simple model of selection. We consider the fitness cost as a function of the density of oncogenes and the tumor suppressor genes. Yeah, it's a very a simplified model because there may be other factors affecting the selection pressure, such as also the ploidy and other factors. Uh, and also you mentioned the introduction of drugs. I think these external factors may also affect their fitness. I think that can be incorporated to improve the model in future.
Okay. Great, great.
Mm.
So it looks like we have several questions here in the chat, Dr. Lu. So from Kisheng, okay. The question is, did you also model the probability of different mutational events? For example, single nucleotide substitution that may happen more frequently than structural variations?
Yeah, here when we focus on one types of mutational events, such as when we focus on SVs, we didn't consider single or nucleotide substitution. Yeah, I think it would be good to integrate the different mutational events. Yeah, in future to get a more comprehensive model, but it also increase the complexity of the modeling. So currently we just focus on one type, like a structural variant on copy number alteration.
Right. I think that's a very important question. I agree. Because in lung cancer, we know we have this scoring called tumor mutation burden,
Mm,
and they have to incorporate this nucleotide substitutions type of mutation. So that would be interesting if you can incorporate in the future model.
mm.
Okay, Kishan, you okay with the answer, or you want to have follow-up questions? Is it okay? Okay, all right. Okay, so let's go to the next question. Okay. Oh, okay. From Abraham Ido, I think a comment that's he's very interested in your PhD program. So could you maybe briefly elaborate about your program and the focus of your research, Dr. Lu?
You mean the program for recruiting PhD
Yes.
students or PhD
Right,
studentship
for PhD
program?
student. Yes.
Oh, I see. Yeah, yeah. Currently, I'm still trying to applying fundings for PhD studentship because in UK we need some external funding to support PhD students. So I know there are some fellowship like I know there's a CSA scholarship from China, and mm, uh, and sometimes there may be some internal studentship from our faculty, but currently it's still unknown. And also there may be some collaborating PhD studentship between University of Surrey and other universities. There's a recent one between University of Wollongong in Australia. Um, yeah, it's a bit difficult to have the fundings for PhD studentship for now. Mm.
But
And
apart from the funding, I mean, your research area itself, I mean, would you mind to elaborate a bit more? Your
like
focus,
my refocus on
your
my
research
research.
focus.
Yeah. Currently, the my research is keeps focusing on the cancer genomics and the chromosome instability. So yeah, as you can see, currently the model is still quite simple. There are still lots of factors that can be incorporated to further improve the model and also to apply it in more scenarios like the drug resistance and yeah, like the drug, like what you mentioned just now of the cases with and without drug and many other situations. And also also interested in um other areas like I'm yeah, I'm currently also affiliated with the AI Institute at University of Surrey, so I'm also trying to apply and develop some machine learning approach
Mm,
for biological problems like drug repurposing and also like cancer driver prediction. And also I'm also interested in the genome graphs. This is also related to structural variants because of the structural variants, they may form very complex cancer genome graphs. And also previously I also worked on microbiome and in the microbiome, they also have this pan-genome graph concepts. And yeah, I'm interested in this genome graphs and related problems.
right. Great.
Mm.
So anybody in the audience who's interested in Dr. Lu's research, write her an email, express your interest. All right. Okay, next question from Hilya. So thank you for an interesting presentation, Dr. Lu. And she asked, are there potential application for your modeling approach beyond cancer research, such as in other disease associated with chromosomal instability?
Yeah. I think so. Yeah, because actually the model approach is quite general. Currently, it's applied to the cancer genomics data, but it can also be applied to other non-cancer disease. I know there are other disease also have this chromosome instability, maybe not so much as in cancer, but there are some like some neurodegenerative disease may also have some large copy number alterations. Yeah, I think it's applicable. Mm.
Right. Oh, yeah, this is because some congenital diseases, they have probably some increased number of copy numbers, yeah.
Mm.
Maybe repeats. Yeah, that's interesting to model as well. Great.
Mm.
Um, I want to remind everybody to don't forget to complete the survey. Yeah, so I put the survey in the chat just in case that while we follow up with the questions. Don't forget to fill up the survey. Okay. Any other question? Anybody want to have a direct question to Dr. Lu? You can raise your hands and open up your videos. I have a lot of questions. So I have no short of questions. But okay. While waiting for other question, let me ask you this one, Dr. Lu.
Mm,
Usually in cancer, we divide it neatly in stages. Yeah, stage
yeah.
0 means no cancer, stage 1,
Mm,
stage 2, stage 3, and the metastatic
mm,
one, stage 4. In
mm,
your understanding the literature or probably experience in the research, do you think cancer actually follow this kind of neat evolution, or actually they're entirely different disease? So for what I'm trying to say is that stage 3 is not actually a product of stage 2 or stage 1.
mm,
I mean, the cancer can just jump to stage 3 disease. Is it like that,
mm,
or which way? I mean, can modeling actually answer this kind of questions?
mm. I think it's yeah, you mentioned this it's like some gradual evolution or punctuated evolution, something like this
Mhm.
is what I actually.
Right,
Yeah,
right.
I think it's yeah, I think it depends. I know for cognitive alteration sometimes there can be this chaotic events in one cell division, like the chromosomes, and yeah, and this can be seen as punctuate evolution. It's like some sudden change point, and then the cancer may cancer cells may be more uh, may have more advantages to grow. And but in term but I think there's also this selection pressures. If
Mm.
there are too much uh changes, there may be disadvantages to cell. There may be some threshold,
Right.
and also I think the the classification of stages is also a bit subjective.
Mm.
Uh, maybe I can say that. Um, but anyway, I think there's a size from the normal to the disease, and for some I know for some cancer, they usually are detected at a late stage because it's very hard to detect them early given very limited evidence. But actually, I think there are something happening in the cancer cells. If we can find some biomarkers or evidence, I think we can get the signals for the cancer development. I think yeah, I think maybe there are some sudden change, but I think it's still a gradual process. Mm.
Right, right. Yeah, because I'm
Mm,
wondering maybe if it's possible to
mm,
look at a patient, for instance, and follow
mm,
the evolution. So say the
mm,
patient had initially stage 1, get that
mm,
biopsy of stage 1 disease, look at the structural
mm,
variation or copy number
mm,
variation. So as now
mm,
the patient moves to stage 4, take the same
mm.
samples again and maybe trying to compare. I mean, is it the right way to design this kind of experiments to compare from the same patients to really understand whether actually the cancer actually really move in stages or
Yeah. I think that's ideal if we have those kind of longitudinal data sets.
Mhm.
Yeah, one issue is I think it's a bit hard to get the samples from patients, especially for solid tumor tissues. I know currently the liquid biopsy has been developing very fast, like
Hmm.
the blood samples. I think
Right.
using those samples, it will be more easier to get the samples at different time points, and then we can use those samples to get a better idea of how cancer evolve over time.
Hmm. Right. Yeah, you're right. I mean, liquid biopsy is very important and also in lung cancer, and also
Mm,
all target therapy, right? For
mm,
instance, one of the target, say EGFR, tyrosine kinase
mm,
inhibitors,
mm,
one of the resistance mutations actually is a MET amplification. So obviously, it's
mm,
the copy number changes actually
mm,
induce resistance to the drug.
mm.
Okay. We still have about 4 minutes, everybody. So please feel free to ask Dr. Lu. Otherwise, I'll be asking you again some questions.
Yeah,
Okay.
yeah. Mm,
Um, please don't forget to fill up the survey. Yeah? I mean, it's very important for our Asia Pacific Bioinformatics Network. Actually, it's a great organization for all of us to share and to network very cutting edge information that we are getting here. So it's very important. Give your comment in the survey. Another question, Dr. Lu.
Mm,
So you know, some cancer cells, they do have, say, HER2 amplifications. This obviously is a copy number changes, right?
mm,
But as far as I know, when it comes to this, say, HER2 amplifications, and when
mm,
you grow them in culture, generally they're
mm.
quite stable. I mean, they don't lose or even gain more of the HER2. How could we explain it? Because I just worry that
Mm.
from the model you mentioned, it looks like it just keep on changing. But from the empirical or from the experience in cell culture, generally the cell lines are quite stable. Is there actually a way to explain it?
Oh, I see. Yeah, yeah. I think the cell lines and the real tissue are really different because of the external factors. Mm. But yeah, I think it may depend. For example, I mentioned one study using the breast cancer cell line. Actually, they found there's ongoing copy number changes, like the copy number out rate had been changing even for some supposed to be identical cells. Um, yeah, maybe I think for the HER2 amplification, it's uh, yeah, there are some different factors affecting the amplification. I know for some oncogene amplifications, they may amplify in the form of EC.
Is it me or I think the connection? Yeah.
Yeah, it's mm, mm, I'm not sure if I know you have this the cell line culture, and there are also like the 3D, like the organoid culture, maybe slightly different if
Mm.
we
Right. Yeah, I mean,
mm,
uh, I would agree that ideally you should get the xenograft or
mm,
organoid from patients, because sometimes
mm,
when you look at the cancer cell lines, they are not,
mm,
I mean, they've been involved anyway, right? Because they've been selected to grow in vitro. So maybe
mm,
very different from the original ones, right? But also
yeah,
getting organoid from patients is not easy. It's not, it's very challenging to grow them in culture. But yeah, I mean, of course, these are limitations, but the way you explain it is very good, that we can get some data to do some modeling.
yeah,
All
yeah, yeah,
right.
yeah, yeah. It's also good to model the data to see what are the differences. Mm.
Yeah, and also technology now we can do single cell sequencing is also quite important, right?
Yeah, yeah, yeah. That's true. Mm,
Although sometimes I kind of wondering because in real world, in patients, the cells grow in environment of lots of immune system. So it's
mm,
also interesting to look at the dynamics, how the presence of immune systems actually also can
mm,
direct the evolution, the clonal evolution of this cancer, because they have to, somehow escape from the immune
mm,
system, right? So definitely they have to, maybe shuffling their genomes to get, to survive or to evade this immune system. I
mm.
don't know whether it could be something to test in future.
Yeah, yeah. I think that's definitely worth looking at in future. Mm.
Right. Okay. I think our time is up. So I guess there will be no other questions. Thank you very much, Dr. Lu, for entertaining us. Definitely is a new information also for me that it's possible to do some mathematical modeling. I'm very bad in math, but I mean, you presented quite nicely. Thank you so much. All right, everybody. Thank you so much for joining us. And hopefully we're going to see you all again in the another exciting session of the Asia Pacific Bioinformatics Network. Thank you.
Thank you. Bye.
Oh, don't forget, it's going to be, this is a pre-recorded. Just go to the AppBioNet YouTube channel, yeah? Um, what is well, the address, just maybe Google it, you'll find it, okay? For anybody who want to learn more, you just look at the recording. Okay. I think that's it for today. Again, I'll see you all again. Bye.